Before starting with the thyroid and parathyroid gland, the microscopic structure, okay. First, we'll see the brief introduction about the thyroid gland and parathyroid gland, and then I will start with the histology, okay. So, see, what I have observed is most of the student, whatever I am mentioning, everything you are writing in the practical notebook. You should not write all those things. Here I am giving you the introduction, though you have not studied the thyroid gland. Okay, that's why I am giving the introduction, then I am going to the histology. It doesn't mean that in the histo notebook you will write everything. In histology, what you will write? Only the histology of the thyroid gland. Okay. See, this thing I should not explain to you guys. You are enough mature now. I have seen students are writing about gross anatomy, introduction, parts, nerve supply, blood supply. Every day when I am correcting, I used to check all those things. Every time I have to write not required, not required, not required. You have to understand, that's why your practical notebook is becoming that much thick. Okay, so as I said, only the brief introduction about the thyroid gland, where I will show you the parts of the thyroid gland and some important peculiarities. Then directly, as I said, I will go to the microscopic structure of thyroid gland. Okay, so the thyroid is shield-like. Thyroid means, the meaning is shield-like. Okay, endocrine gland having rich blood supply, situated in the lower and in front of the neck. Lower part and in front of the neck. That is the location of the thyroid gland. Now, if you see the thyroid gland, the main function includes control or regulate the basal metabolic rate, stimulate the somatic and psychic growth of an individual, an important role in calcium metabolism. These are some important functions of the thyroid and parathyroid glands of our body. Now, the thyroid gland is placed superficially so that it can be palpated easily. Okay. Next, as I said, there are some peculiarities of the thyroid gland. For example, first peculiarity is it is the only endocrine gland which is situated superficially. Only endocrine gland situated superficially. Rest all the endocrine glands situated deep inside the body. Second important peculiarity is for production of hormone, it is dependent on external source. That means iodine. Without iodine, it cannot produce hormones. This is a peculiarity. Other endocrine glands do not depend on any other source for production of hormones. This is the only endocrine gland which depends on external source, that is iodine, and iodine cannot be produced in our body. That's why it is completely dependent on the external source. Whatever we are eating, from there it is getting the iodine. Next, thyroid is the largest endocrine gland of our body. I hope you know that. जितना भी endocrine glands है in our body, among them thyroid is the largest endocrine gland of our body. Okay, so these are some important peculiarities of the thyroid gland. Next, if you see the thyroid gland, what are the parts of the thyroid gland? So two lobes are there of the thyroid gland, two lateral lobes, right and left, and they are connected by isthmus. Okay, occasionally a third lobe appears from the isthmus. Okay, but it is occasional, every time it is not present. Next, there is a fibromuscular band extending from the isthmus to the hyoid bone, that is called as levator glandulae thyroideae. Okay, descend from the body of the hyoid bone to the isthmus of the thyroid gland. Okay, here you can see the diagram. So in this diagram, this is one lobe of the thyroid gland, this is another lobe of the thyroid gland. So two lateral lobes are there. Now these two lateral lobes are connected by isthmus. Occasionally a third lobe is present, that is called as pyramidal lobe. Okay, and you can see a fibromuscular band called as levator glandulae thyroideae is attached to the isthmus from the hyoid bone. Okay, next, so this is the structure of the thyroid gland, overall structure. Next, in the gross, you have to study about the surfaces, borders of each and every lobe, and in case of isthmus also surfaces are there. Okay, that you have to study. Then about the capsule of the thyroid gland, okay, nerve supply, blood supply, all those things in the gross part. Okay, so though you have seen the thyroid gland, you have seen the parts of the thyroid gland. Now directly we will go to the histology. Okay. Now suppose you have taken one section over here, or one section over here, or one section over here. Okay. Then what you can see inside the gland? I hope you can see the capsule. Can you see the capsule of the thyroid gland? तो thyroid gland में भी true capsule, false capsule रहता है. That will be discussed later on in the gross lecture. But for the time being, please keep that in mind. In case of thyroid gland also, true capsule and false capsules are present. So in the histo notebook, you have to write it right from here. Okay, the histology of the thyroid gland. Now, do you remember if any gland or any structure where capsule is present, then what will be the structure? If you have studied about the salivary glands, in that we have seen that salivary glands, they are also having capsule. When we have studied pancreas, at that time we have seen capsule is there. Same story is repeated. Here also you can see thyroid gland is covered by dense irregular connective tissue capsule, which is derived from investing layer of deep cervical fascia. After that what happens? From capsule what will go inside the gland? Connective tissue septa. Here also connective tissue septa is extending into the gland and dividing into small lobules. Okay. So you can see on microscopic examination, each lobule of the gland is made up of aggregation of follicles. So septa extending from the gland from the capsule divided into lobules. Okay, and the structure is same, almost same for thyroid gland also and for parathyroid gland also. Next, if you take one particular lobule, then what you can see inside the lobule? In case of thyroid gland, not parathyroid, in case of thyroid gland, these are made up of follicles. Okay, multiple follicles are present. So one lobule, what you will get inside the lobule? Multiple follicles. So what is there inside the follicle? Any idea? Something will be there inside the follicles. So follicular cells are present inside the follicles. Okay. So see, mainly the parenchyma of the thyroid gland is made up of follicles. These are the spherical masses called thyroid follicles. The size is 0.2 to 1 millimeter in diameter. All the follicles are not having same size. The size of the follicles are different. Next, inside the follicles, what is present? As I said, follicular cells are present inside the follicles. Now it is lined by which epithelium? Simple cuboidal to low columnar epithelium. Now here also the lining epithelium changes. Every time you are not getting the same lining epithelium. That you have to understand. When we are getting simple squamous, when cuboidal and when columnar. Next, at the center of the follicle, what you will get? You will be getting gel-like acidophilic substance, and these are called as colloid. Okay, gel-like acidophilic substances are present. Okay, and the appearance is homogeneous, pink color appearance hoga. Okay, and these are called as colloid. Now these colloid are rich in glycoprotein. Okay, and these are precursors of T3 and T4, the hormones which are secreted from the thyroid gland. Okay, mainly ये जो colloid है, these are made up of thyroglobulin. It is a type of glycoprotein. Okay, now what happens when it intakes the iodine, then iodine will be added to that thyroglobulin, and then finally we are getting the T3 and T4. Okay, this is the way it is produced. That's why inside the follicle you will be getting a homogeneous pink color substance under H&E slide, and those are called as colloid. तो क्या रहता है colloid में? What is present in the colloid? As you can see, gel-like substance. Now this gel-like substance is made up of thyroglobulin. It is a type of glycoprotein. Next, under H&E section, the follicular cell shows slightly basal basophilic cytoplasm and spherical nucleus, and sometimes one or two nucleoli are present. What is the meaning of this? When you will be preparing the H&E slide of thyroid gland, the follicular cells shows slightly basal basophilic. इसका मतलब क्या? What is the meaning of this? Basal basophilic cytoplasm. That means basal part will be darkly stained. Okay, basal basophilic. Okay, darkly stained, or you can say basal part will be will appear purple or pink. Purple or blue in color. Okay, that is basal basophilic. Next, interfollicular space is there. That means in between the follicles the space which is there, that is called interfollicular space. उसमें क्या रहता है? Nerve fibers, blood vessels, lymphatics, all those things are present in between the follicles, and these are called as interfollicular space. ठीक है? Next, in the interfollicular space, another special type of cells are present, and these are called as parafollicular cells. Okay, these are called as parafollicular cells. Now, these parafollicular cells are embedded either on the follicle or in between the follicles. Now, these are polyhedral cells with oval eccentric nuclei. Now, cells single or small clusters, mostly they are in the form of clusters, these cells. Okay. Now, these cells are difficult to identify under light microscopy. Parafollicular cell shows numerous secretory vesicles on their apical part of the cell. Follicular cells secrete two hormones, that is thyroxine T4, triiodothyronin, that is T3, and parafollicular cells secrete a hormone that is called calcitonin. Okay, these are the functions of the two different types of cells present in the thyroid gland. So, what is present in the thyroid gland from the beginning in histology? First of all, connective tissue capsule. It will extend septa into the gland, divided into lobules. If you take one lobule, what is there inside the lobule? Follicles. Multiple follicles are there. Follicles are lined by which cells? Follicular cells. So, what are the different types of follicular cells? Either it may be simple squamous, it may be cuboidal, it may be columnar. Okay. When which cells are there, we will discuss. Next, what is present inside the follicle? Colloid is present. Okay, colloid is made up of what? Thyroglobulin. It is a type of glycoprotein. These are the precursor of T3 and T4 hormones. Okay. Next, in between the follicles, the area which is present, इसका नाम क्या है? What is the name of that area? It is called as interfollicular space. इसमें क्या रहता है? Interfollicular space में what is present? Blood vessels, nerves, lymphatics, all those are present. Apart from this, a special type of cells are present. These are called as parafollicular cells. Okay. Now, if you see the parafollicular cells, these are polyhedral shaped cells. Mostly they are in the form of cluster. Okay. Secretory vesicles are present on the parafollicular cell. Now, follicular cells, they secrete two hormones, that is T3 and T4, also called as thyroxine and triiodothyronin. And parafollicular cells secrete a hormone that is called calcitonin. Now, you have to know that which type of cells are present and when they are present in the follicles. That means the activity of the follicle. So, as I said, follicular cells vary in shape from squamous to cuboidal to columnar depending on their activity. Okay. Now, suppose if a follicle is highly active, at that time what will happen? Columnar cells will be present. An amount of colloid will be very less inside the follicle. Okay. Next, when it is lined by cuboidal cells, it is moderately active and moderate amount of colloid is present. Now, if the cells are follicles are inactive, then it is lined by simple squamous epithelium and abundant amount of colloid are present. Okay. So, these are the different types of cells present inside the follicle. Okay, depending on the activity. Okay. So, follicular cells, already I have told you what is the identifying features of the follicular cell. When you will see the follicular cell under electron microscope or electron microscope structure, apical microvilli are there, abundant granular ER, prominent supragranular nuclear complex, microtubules, microfilaments, all those things can be seen under EM structure. Okay. Here you can see the diagram. Both the diagrams you have to draw in your practical notebook. Both the diagrams. If you concentrate over here, in this diagram you can see the activity of the follicles. As I said, inside the thyroid gland lobule, follicles are there. Okay, so these are the different types of follicles. So this is highly active, this is moderate, and this is inactive follicle. Can you see the cells? So which cells are this? Simple columnar epithelium, and you can see the amount of colloid is very less. Next, which type of epithelium is this? Cuboidal epithelium. Now here moderate amount of colloid is present. And what about this? Simple squamous, inactive. Okay, so that's why huge amount of colloid is present. So this depends on the activity of the follicle. And here you can see the EM structure of follicular cells. The follicular cells is the EM structure of the follicular cells. Now what do you think, how the activity of the follicle is determined, or who is controlling the activity of the follicular cells or activity of the follicle? Pituitary gland. Okay, so which hormone from the pituitary gland? TSH, thyroid stimulating hormone. Okay, that is controlling the activity of the follicles. So you see, activity of the follicular cells are influenced by TSH, thyroid stimulating hormone, which is secreted by pituitary. Now what is the other name of pituitary gland? Hypophysis cerebri. Hypophysis cerebri, it is the other name of the pituitary gland. Okay, so in the exam mostly questions are given as write about hypophysis cerebri, or what is hypophysis cerebri, what are the parts. So make it a habit to understand that pituitary is nothing but the hypophysis cerebri. Next, the functions, T3, T4 hormones, what is the main function? Control the metabolic rate of the body, and in fetal life and in early childhood, this T3, T4 promotes the CNS development and growth. That means psychosomatic growth of an individual, that is controlled by the T3, T4 in during intrauterine life and early just after the birth. Next, calcitonin, which is secreted by parafollicular cells. What is the function of this calcitonin? As you can see, it lowers the blood calcium level. Okay, this calcitonin, what it will do? It will lower the blood calcium level by increasing the calcium deposition in the bones and inhibiting the bone reabsorption by osteoclast. Okay, so osteoclastic activity will be suppressed by this particular calcitonin hormone. Thus, it is reducing the calcium level, serum calcium level. Okay. Now, can anyone tell me which hormone will increase the serum calcium level? Yes, bolo. Parathormone. Okay, PTH, parathyroid hormone or parathormone. Okay, it increases the calcium level. So like this, the balance is maintained. Now here you can see, this is the schematic diagram which you have to draw in your practical notebook. ठीक है, तो क्या दिखाई दे रहा है? What you can see in the diagram? So in this diagram, capsule is not seen, but instead of that you can see the connective tissue septa. Can you see the connective tissue septa, everyone? ये देखो, this is the connective tissue septa. This is the connective tissue septa. So in between the connective tissue septa, you can see this is one lobule. ठीक है, this is one lobule. Now inside the lobule, what are this? ये क्या है? This is connective tissue septa. This is connective tissue septa. Inside that, this is the lobule. So this is one lobule, this is one lobule, this is one lobule. ठीक है. Now inside the lobule, what are these? Follicles. ठीक है. Now inside the follicles, what you can see? Two things are there. First follicular cells and then colloid. So can you see the homogeneous nature of the colloid? Easily it can be identified. Now all the follicles are not having equal amount of colloid. Why? That depends on the activity of the follicle, which is stimulated by TSH, thyroid stimulating hormone. Okay. What else you can see in the connective tissue septa? Can you see the blood vessels? ये देखो. So I hope you can see the blood vessels in the connective tissue septa. Next, in between the follicles, can you see the parafollicular cells? For that you refer to that magnified version, this magnified part. Okay, so see, this is one follicle, this is one follicle. Okay, so here you can see these are lined by follicular cell. So this is another follicle. In between these two follicle, in intra follicular space, here you are getting the parafollicular cells. Okay, तो parafollicular cells का जो identification होगा under H&E stain, it is pale in stain. Okay, and this follicular cells, these are basal basophilic, so indirectly you can say these are darkly stained. So like this the differentiation can be done between the follicular cells and parafollicular cells. Okay, and parafollicular cells, as I said, mostly they are arranged in the form of cluster. Okay, and in this particular space in this particular space you can see the blood vessels are also present. I hope you can see the blood vessels. Okay, so identification of slide of the thyroid is very easy. I am talking during the time of exam. ठीक है, एग्जाम में अगर ये स्लाइड दिया तो इट विल बी वेरी इजी फॉर एवरीवन टू आइडेंटिफाई. Last time, if you remember, I have told you that identification of the slides of urinary system, reproductive system is very easy. Similarly, in head and neck portion, जो thyroid gland है, identification is very simple, very easy. Okay. So this is a low magnification image of thyroid gland. This is high magnification image of thyroid gland. ठीक है. तो low magnification में क्या दिखाई दे रहा है? What you can see? Can you see the lobule? So how you will identify the lobule? In between the connective tissue septa, you have to identify the lobule. Can you identify the lobule? Yes. Once you identify the lobule, see inside the lobule. क्या दिखाई देगा? What you can see inside the lobule? Follicles. So try to identify the follicles. Now when you are looking at the follicles, you have to see the amount of colloid inside the follicle, because under low magnification you cannot identify the lining epithelium, but you can identify the amount of colloid present. ठीक है. So like this you can identify which is active, which is highly active, which is inactive. Okay, based on that you can say which type of epithelium are present. Okay, next parafollicular cells are also present. Okay, now see, these are the parafollicular cells, or also called as C cells. So how we will differentiate between the follicular cells and parafollicular cells? ये देखो, this particular area, okay, here parafollicular cells are present. First of all, parafollicular cells will be present in the interfollicular space. So suppose this is one follicle, this is one follicle. In between these two follicle, this is the space where the parafollicular cells are present. ठीक है. तो इसका जो साइटोप्लाज्म होगा, that is pale staining. Okay, and जो follicular cells है उसका जो साइटोप्लाज्म है, it is basophilic, or you can say darkly stained. So like this easily it can be differentiated. Okay, so I hope it is clear about the thyroid gland. Okay, what is the next? Parathyroid gland. Where the parathyroid glands are situated? Yes, on the posterior border of the thyroid gland. How many parathyroid glands are there? 4 parathyroid glands are there. Only this one. This one and wait, this one. See, one thing I have added, that is the formation of the T3 and T4. Okay, but this you don't have to add, or you don't have to write in the practical notebook. Okay, this you will study in physiology. The T3 and T4, how they are produced, what is the mechanism. Okay, though it is there, but you don't have to write in the practical notebook. So parathyroid glands are two pairs of small endocrine glands situated on the posterior border of the thyroid gland within the false capsule. Okay, now here also each gland is having connective tissue capsule from which septa extend into the substance of the gland, same like the thyroid gland, but there is a difference. And what is that difference, that you have to understand. Now here you can see within the gland network of reticular fibers are there. Okay, many fat cells are also present within the stroma. Next, the parenchyma of the cells are arranged in the form of cords, and in between them the spaces which are there will be getting the numerous sinusoids. Okay, so sinusoids are specially present in case of parathyroid glands. Here you can see, this is the posterior view of the thyroid gland. Okay, this is the posterior view of the thyroid gland, where you can see the parathyroid glands are situated on the posterior border of two lateral lobes of the thyroid gland. Now in parathyroid gland also, already I mentioned connective tissue capsule, septa will extend, but when the septa is extending, the lobes which are formed, okay, the lobules which are formed inside the parathyroid glands, these are not complete, these are incomplete lobules. Now the parenchyma are richly supplied with sinusoids. Okay. Now in the parenchyma, follicles are not there. Instead of that, we are getting two different types of cells. What are these cells? Chief cells and oxyphil cells. These are the two different types of cells present in the parenchyma of the parathyroid gland. Okay. Chief cells and oxyphil cells. So they are in the form of cords, and it is having rich, or you can say more number of sinusoids. Now what about the chief cells and all, also called as the principal cells? Now they secrete hormone. What is the name of that hormone? That is called parathyroid hormone. And the number of chief cells or principal cells are more as compared to the oxyphil cell. Under light microscope, if you see, chief cells are small polygonal. The diameter is approximately 7 to 10 micrometer with centrally located nucleus. They have dark staining cytoplasm. Okay, and this dark staining cytoplasm is based on the parathyroid hormone content, how much the hormone is present. Okay, and depending on the quantity of glycogen and lipofuscin, lipofuscin is a yellow color pigment. Depending on these two material, the principal cells are divided into three categories. What are they? Light, dark and clear cells. And under electron microscope, it shows rough ER, abundant rough ER, Golgi apparatus and secretory vesicles. Okay, these are the properties of the chief cell. Chief cells are the most important cells in the parathyroid gland. Why? Because they secrete parathyroid hormone. Okay, or also called as PTH. Now what is the function of the PTH? It tends to increase the blood calcium level. Now how it will increase the blood calcium level? What are the mechanism behind increasing the blood calcium level? Increase hota hai, theek hai. How it will increase? Yes. Demineralization of. Okay, that means it will increase the osteoclastic activity. Next, it will increase the absorption from gut. Whatever you are eating from the gut, it will increase the absorption. Then again another mechanism is there from the renal tubules. Okay, from the renal tubules it will increase the absorption, from the gut it will increase the absorption. Okay, so like this it will increase the amount of calcium, blood calcium level. So parafollicular cells, it secretes calcitonin, it will decrease, and this parathormone, it will increase the calcium level. Like this, it is balance is maintained. Next, another type of cells are there. These are called as oxyphil cells. Okay, if you see the oxyphil cells, first of all, the number of oxyphil cells are less as compared to the principal cell. Oxyphil cells are large in shape. Okay, if you see the staining properties, these are lightly stained. Okay. Now lightly stained, what is the meaning of this? Oxyphil cells have abundant deeply eosinophilic, or you can say acidophilic cytoplasm because of large number of mitochondria. Okay, eosinophilic cytoplasm मतलब उसका appearance कैसा होगा? Pink appearance होगा. Okay, that's why I have told you pale appearance will be there. Next, these particular cells do not show any secretory vesicles. Okay, now this particular oxyphil cells which are present in the parathyroid gland, they don't have any function, any secretory function. If they are having any function, still it is unknown. So cells do not have any secretory function, and the function of the oxyphil cells are still unknown. Okay. Now these are two diagrams. Among them, this you have to draw, only this one you don't have to draw. Now this is the schematic diagram of parathyroid gland. तो क्या दिखाई दे रहा है इसमें? What you can see in the diagram? I hope you can see the capsule. ये देखो, this is the connective tissue capsule. In the connective tissue capsule you can see blood vessels are there, some adipocytes. Okay. Next, from the connective tissue capsule you can see these are the septa. Now here the septa which is extending into the gland, it cannot divide the gland into complete lobules. Lobules are there but it is not complete. Incomplete lobules are there, and I hope you can see in the diagram. Okay. Next, you can't see follicles over here. Instead of that, what you can see, two types of cells are there. What are those cells? Principal cells and oxyphil cells. How you will differentiate principal cell and oxyphil cell? Number of principal cells are more, number of oxyphil cells are less. Okay. Now here you can see these are the, ये जो cells है, what are these cells? These are chief cells. Okay. Now see the appearance of chief cell under H&E stain. What is the appearance? Darkly stained. Okay. And see, these are the oxyphil cells. Can you see? These oxyphil cells are larger as compared to the chief cells. Okay. And the numbers are also less. Next, if you see, their cytoplasm is acidophilic. That's why you can see pink color appearance. Okay. Like this you can differentiate. तो ये जो सेल्स है, चीफ सेल्स है, इसका फंक्शन क्या है? टू सिक्रेट पैराथायरॉयड हार्मोन. पैराथायरॉयड हार्मोन का फंक्शन क्या है? इट विल इंक्रीज द कैल्शियम लेवल. ठीक है. एंड व्हाट अबाउट द ऑक्सीफिल सेल्स, इसका फंक्शन क्या है? नो फंक्शन. ओके. नेक्स्ट, यू कैन सी सम एडिपोसाइट सेल्स आर ऑल्सो प्रेजेंट. सी सम एडिपोसाइट सेल्स आर ऑल्सो प्रेजेंट, एंड जस्ट adjacent to the chief cells you can see these are the blood vessels, or you can also say these are the sinusoids present. Why sinusoids are associated with this particular type of cells? Because parathyroid hormones are secreted. Hormone secrete होने के बाद कहाँ पे जाएगा? Where it will go? It will go to the blood stream directly. That's why the blood vessels are more as compared to the chief cells, means near the chief cells. Okay. Next, this is the low power magnification. Okay, low magnification of parathyroid gland. So what you can see, these are the blood vessels. Okay, you can see the oxyphil cells. See, these are the oxyphil cells and these are the chief cells. This is the connective tissue septa. Okay. So slides of thyroid, parathyroid is not that difficult. It's very simple. Okay. Just you have to know, especially in case of thyroid gland, you have to know the activity of the thyroid gland, when it is highly active, moderate, and when it is inactive. Based on that you have to know the cells, their secretion and their function. That's it. Okay, so now we'll go to the lab and we'll see the slides.